Hello, this lecture is on chemical reactions and the function of enzymes for students taking Anatomy and Physiology 1. Chemical reactions in the human body occur as atoms and compounds interact and collide with one another. The collisions cause formation, rearrangement, or the breakage of chemical bonds. Collectively, all of the chemical reactions that happen in your body are referred to as metabolism. We represent a chemical reaction using a written equation. So the arrow symbolizes the course of the reaction from beginning to end. And the beginning compounds, the starting compounds, are called reactants. They are on the left side of the arrow. And after the reaction is done, the products are written then on the right side of the arrow. For example, the reaction that happens in the box occurs when methane, CH4, and oxygen, O2, interact and then recombine in such a way that carbon dioxide and water are formed. Now there's many different ways to categorize and talk about chemical reactions. The first way that we're going to do this is related to the use or release of energy. So what energy is the capacity to do work. That comes from physics, really. So some chemical reactions require that we put in energy. And I've got energy symbolized with this yellow star here. When energy is required for the chemical reaction to proceed, we call it endergonic. If instead energy is released when the chemical reaction occurs, we call it exergonic, to release the energy. Now there are different states of energy, maybe you call them different forms of energy. In general chemistry and general physics, if you take it, you'll talk about different states of energy. One state is potential energy. This is stored energy that can be used later. In our bodies, this energy is stored in chemical bonds. So potential energy in the body is found in chemical bonds. Kinetic energy is a different state, and this is movement energy, and that's the release of energy. Now we can convert one form of energy to another when bonds are either formed or broken. So when a chemical reaction happens where something has been broken down, so we have a large molecule here broken down into two smaller molecules, we know that potential energy has been converted to kinetic energy because the bonds have been broken and energy has been released. You could also call this an exergonic reaction. I'm sorry this is yellow, but it's faster that way. Another way that we convert from one form to the other is our bodies take small compounds and put them together to form a larger compound, and bonds have been formed to make that larger compound. In this way, these beginning or starting molecules are moving, and that's kinetic energy. When they meet up and a bond is formed, now the energy is stored in those bonds, and we call it potential energy. This is typically an either way, endergonic reaction, because the input of energy was required. Another way to talk about chemical reactions not necessarily related to energy has to do with what happens with the reactants. So in some chemical reactions, the reactants are broken down. That's catabolism. In other types of chemical reactions, the reactants are smaller, but the product is synthesized. That's an anabolic reaction. Often these two will happen kind of sequentially, so that what you end up really getting is, in sum total, an overall reaction that is an exchange reaction, where two reactants have been broken down or catabolized and two products are formed or anabolized. An example of an exchange reaction is called an oxidation-reduction reaction, and this has to do with just the transfer of electrons from one reactant to another, and I'll show you an example so that makes more sense. Another way we can describe chemical reactions isn't necessarily based on what happens to the reactants, but instead defines the fact that the reaction is reversible and it can happen in either direction, and which direction it occurs in depends on a number of factors, and we'll look at the major ones. So here are some examples of the different types of reactions I just mentioned. A catabolic reaction that takes place in the body occurs in the liver when glycogen, which is a carbohydrate stored in the liver, is broken down into glucose. This happens in between meals. So when glycogen is broken down, individual glucose molecules are the product. This happens to be exergonic as well, because energy has been released. An example of an anabolic reaction occurs when we build proteins. Think of a muscle enlarging when you do weightlifting. Individual amino acids are put together to make a larger molecule known as a protein. So that's an example of an anabolic reaction. Here's an example of an exchange reaction. So essentially catabolism and anabolism both happen. So bonds will be broken. So here are two reactants, hydrochloric acid and sodium bicarbonate. They're going to be broken down and something else is going to be rebuilt. So this will be these are what will be catabolized, these two reactants, and the products will be formed. So there's also an anabolic component. Okay, so for example, this hydrogen from hydrochloric acid is going to combine with the bicarbonate, and so this is the product that you get. They're just swapping partners, really. And then the chlorine will go to the sodium and you'll get table salt. So that's an example of an exchange reaction. It's also shown in this box over here, where you can see that partners are just switched, and I used the same color coding in the reaction I just talked about, but the red reactant is with the orange one, and it's going to change partners and go to be with the blue one. Okay, and the orange is going to go partner with the green one. So it just flips. An oxidation reduction reaction is interesting because you start with two reactants. Here's one compound, it's just generically called A, and here's one generically called B. Some reactions don't really well, we'll talk about that in a second, but in this reaction what happens is electrons are transferred, a bond usually breaks, but electrons are transferred to the other starting reactant, but you get a completely different product. So B is not necessarily the same as it was when it started. And of course A isn't the same because it lost electrons. So I want to talk about oxidation reduction because this relates to what we talked about with ion formation. So remember, when a compound, molecule, atom, whatever it is, loses an electron, it becomes oxidized. So A is oxidized because it loses electrons. B is reduced because it has gained electrons. This whole concept of oxidation reduction reactions is very useful for making an energy storage molecule called ATP. And when we talk about how cells make ATP, we'll come back to oxidation reduction reactions. So the last type, or I guess category of reactions, has to do with this one, reversible reactions. Now, you could be an anabolic reaction and still be reversible. So just because you're reversible doesn't discount these other categories. What a reversible reaction is any reaction, regardless of whether it requires energy, builds something, breaks something down, that can happen in either direction. So a reversible reaction can happen going to the left or to the right. And usually an equilibrium is achieved when the reactants and products in terms of quantity are equal. So for example, if I have this reaction where the compound AB gets broken down into two subunits, A plus B, an equilibrium will be reached so that the quantity of the big AB equals the quantity of the subunits A, B. Okay, one place that this happens that's really important in the human body, this concept of a reversible reaction and reaching equilibrium, is in the respiratory system. So all of our cells make CO2, and this CO2 is taken up, it goes into our bloodstream, and in our bloodstream we have water. So CO2 is always exposed to water, and it combines with water to form carbonic acid. Well, that carbonic acid spontaneously dissociates into these two ions. So the more CO2 there is being produced by your body, the more likely the reaction is going to happen towards the right. Notice what that does in terms of hydrogen ion production. That's supposed to be an arrow. So hydrogen ions, the amount will increase when you have a lot of CO2. But let's say, for example, your body has excess hydrogen ions for a different reason. You drank a lot of coffee, you drank a lot of Coke, your urinary system isn't working very well, and so your urine doesn't contain hydrogen ions, so your body retains hydrogen. This reaction can happen in the opposite direction because hydrogen ions have gotten quantitatively so high in the body, and they get converted through this reaction into water, and carbon dioxide is produced. Eventually an equilibrium is reached so that the amount on one side here is equal to the amount on the other side. So this concept of reaching equilibrium is related to something known as the law of mass action. So in our reaction that we just looked at, if the concentration, and this is my first bullet point here, if the concentration of carbon dioxide is a lot greater than the concentration of this carbonic acid here, so this is super high and it's low here, which way do you think the reaction will occur? Both of these two steps. I'm sure you know. To the right. But if instead what you had, I should probably erase all that. Okay, if instead what you had was that hydrogen was really high compared to the amount of CO2. The reaction is going to happen in the opposite direction, and this is due to the law of mass action and trying to reach equilibrium. Now that's direction. That dictates the direction of reversible reactions. It doesn't say anything about the speed with which it can happen. The rate at which a reaction happens depends on how frequently molecules interact with each other, collide with one another. And this will, of course, this rate will be increased when there's a greater or higher temperature, because molecules move more when there's heat. Heat's a form of energy, and so they move more. There's also another thing that can cause a reaction to happen quickly, and that is a catalyst. So in the body, catalysts are referred to as enzymes. So a catalyst here, they temporarily bind a reactant and put the or more than one reactant, many reactants, can be it's usually two. The catalyst holds them in a conformation that makes it quicker for the reaction to proceed. So in our bodies, biologically speaking, these catalysts are called enzymes, and they're proteins. That's what they are. Now they can be reused over and over again, and what they're really doing is decreasing how much energy is needed for the reaction to occur. So first we're going to just look at how an enzyme works, and then we'll talk about the energy requirements. So here's an example of a reaction that's taking place using an enzyme. So let's first just look generally at the reaction, and I'll put that in yellow. We've got here sucrose, which is just table sugar. In your body, that can be broken down, and also it's these two molecules, that green and kind of orange looking compound. I should use something different. Use this one. This is our reactant. That is the reactant right here. It gets broken down into these two things, glucose and fructose. Okay, that's our reaction. That's catabolism. Okay, now that it does release energy when something's broken down, a bond is broken and energy is released. However, something I haven't told you yet is even catabolic exergonic reactions do require a little bit of energy input just to get started. And what the enzyme does is hold the sucrose in a favorable conformation or shape so that the reaction's more likely to occur. And the name of the enzyme that can bind to sucrose so that this occurs is called sucrase. Oops, that's not very good. Better take that off. Okay. Sorry. Is sucrase right here. So sucrase is the enzyme that facilitates the breakdown of sucrose. And they bind the reactant and the enzyme, and then the products are released. And now the enzyme can be reused. Okay, so the enzyme can go bind another sucrose. Now let's talk about that energy requirement business that I referred to before. This graph shows you a catabolic reaction. So you have a reactant that gets broken down into smaller subunits. The process of that happening releases energy. So energy is released. That's what this shows. And the amount of energy that is released is about, okay, I'll use a different marker, is about this much from here to here. If you could quantify how much potential energy is stored here and how much energy is released, the difference, that's what's released. But to get started, what has to happen is an input of energy. So you have to put in a little bit of energy, and then you get a big payback, and a lot of energy is released. So that's typically how it works without an enzyme. And this is for exergonic, catabolic reactions. You still have to put a little bit of energy in. You have to supply a little bit. It's called activation energy. The activation energy. Now, when there is an enzyme present, we say that the reaction is catalyzed. And now the reactant binds to the enzyme, and the reaction proceeds. Notice that the amount of energy required to get the reaction to proceed, the activation energy, is much smaller. So you get a much bigger payout proportionally. So that's an example of how an enzyme helps a reaction proceed. Now, it is true that enzymes also help endergonic reactions occur. That's supposed to be an E. They also help endergonic. But this particular slide is showing you how they even help exergonic reactions take place. The only thing I have left is just a few things to show, a few examples of enzymes and how they work. They're just like real examples versus theoretical. So here's one that you'll come across later. We have a few reactions. Here's 1, 2, 3, 4 reactions occurring. And one thing I wanted you to notice is that there's a different enzyme for each chemical reaction. So in this case, we have a reactant that looks like this. You don't have to memorize that crazy name. And it binds to enzyme number 1. Okay, that's what it does. It binds to enzyme number 1. And eventually you're going to get a product, right? And when it's broken apart, here are the two products. And now this enzyme can go bind another. I think I misspoke previously. Hold on just a second. Let me see. I'm sorry, I did misspeak. This slide's a little different than what I expected. So this particular slide you can ignore largely. We'll just take it out, but it's just showing you that an enzyme can be reused. That's all it is. Let's go to this particular example of enzymes functioning. So here's an enzyme that binds to a certain reactant. And when an enzyme binds to a reactant, for some reason, we don't call it a reactant, we call it now a substrate. So a substrate is the reactant that binds to an enzyme. There are various things that can help an enzyme function. Here are two of them, coenzymes and cofactors. So a coenzyme is organic, which means that it is carbon-based. That's what organic means. It doesn't mean there's no there's no chemical treatment. It means carbon-based. Cofactors, on the other hand, are inorganic, and they're usually some kind of a metal, like calcium. So, this is our substrate, this red thing. It's a reactant that's going to bind to this enzyme. It can only bind, though, if this coenzyme or cofactor first binds to the enzyme. It makes the substrate binding site better so that the enzyme functions better. Here's an example that you'll encounter of coenzyme function. The substrate or reactant called ethanol, common ethanol, alcohol, gets turned into ethanal when it is catalyzed by this enzyme called alcohol dehydrogenase. So alcohol dehydrogenase binds to ethanol, I'll use this one, binds to ethanol and produces ethanal. But notice there's something else in there, this little green molecule thing. Okay, that's a coenzyme called NADH, and it helps the whole reaction take place. In this instance, what it really does is it takes a hydrogen away from ethanol. Notice it's got a hydrogen on it now, NADH. It also takes an electron. Here's an example of cofactors functioning to help enzymes. It occurs in blood clotting. So in your plasma you have several clotting factors. One of them is clotting factor number 9, and it eventually is going to help the whole process of a clot form. And one of the reactions that happens to form a clot is that this clotting factor 9 has to be activated. So this is activated clotting factor 9. In order for that to happen, there's an enzyme, there's an enzyme and calcium acts as a cofactor for the reaction to take place. If you did not have this cofactor calcium present in your blood, this clotting factor 9 couldn't be activated and you wouldn't form a blood clot. So what we've been looking at are chemical reactions and how to read a chemical equation, a reactant to product. And we learned about the fact that sometimes a reactant is called a substrate when it binds to an enzyme. Okay, that's the gist. We also talked about catabolic versus anabolic reactions and energy requirements. But what you will see in different parts of ANP1 and ANP2 is a series of chemical reactions. One reaction, then another, then another, and then another. This happens in blood clotting in particular. So what I wanted to let you know is that every individual reaction will use a separate enzyme because the substrate will be different for every reaction. So here's a picture, or flow chart of the formation of this compound called pyruvate. It forms from glucose in your cells. Your cells break down glucose and make pyruvate. It turns out there's 10 reactions, and these 10 reactions each have a different enzyme shown in red. Hexose kinase, phosphoglucose isomerase, You are not memorizing this. I don't want you to memorize it. But I do want you to see that for every single step there's a different enzyme, and that's because for every single step there's a different reactant. So in our first step, the reactant is glucose, so it fits in a certain enzyme. For step number 2, the reactant is glucose 6-phosphate. It wouldn't fit into hexose kinase. It needs its own enzyme. And that happens on down the line. So this is an example of what we call a biochemical pathway. You aren't going to have to memorize biochemical pathways, but the concept of an enzyme being specific for one chemical reaction is important. That's all for chemical reactions. Thank you.